This is Amanda Welch, welcoming you to this Bite-Size Bio web seminar, which today is sponsored by Bio-techne. Today's presentation is titled Tips and Tricks for Western Blotting and Antibody Selection and is being presented by Paul Joseph from Bio-techne. Paul has spent many years in both industry and academia, both training, giving seminars, and troubleshooting common challenges experienced by life science researchers. He has been associated with several areas of research, such as epigenetics, apoptosis, and cell signaling, as well as techniques such as Western blotting, protein multiplexing, flow cytometry, and genomics. Paul Joseph earned a bachelor's degree in microbiology from the University of Massachusetts at Amherst, and later went on to earn a master's from the Department of Infectious Diseases and Microbiology at the Graduate School of Public Health at the University of Pittsburgh. Now, as always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Paul at the end. So now, over to you, Paul, for the presentation.
Amanda, thank you very much. I appreciate that and your organization, and I also want to thank my inside colleague at R&D Systems, Dave Finkel, for all of his hard work to get this to fruition. And I want to thank you, the researcher, for taking the time out of your busy day. I know you've got a lot on your plate, and you've got experiments to run, and meetings, and grant writing, so we really appreciate you taking the time to attend this. I want to just quickly, before we launch into actual troubleshooting and optimization of Westerns, introduce to you Bio-techne. And usually when I'm giving this talk live, I poll my audience and I ask, well, how many people have heard of the name Bio-techne? And surprisingly, I'm getting a lot more people that recognize this name. This was really a result of a reorganization on the part of, mainly R&D Systems. And the goal, we weren't purchased by anybody. We simply reorganized to better serve you, the researcher. And how do we better serve you, the researcher? We innovate, we offer new technologies and assays, and continue to provide gold standard and trusted reagents for your precious experiments. Now, I mentioned the word trust, and I think it's a huge word when it comes to selecting vendors, because at the end of the day, if you're using inferior quality, you're going to have to repeat that experiment, which means more time delays, more money spent, more time delays in publishing your drug pipeline, and so on. So within the Bio-techne umbrella or family, we have, of course, R&D Systems is the gold standard for recombinant proteins. The first protein company in the entire world with TGF-beta 30 years ago, and today most highly published immunoassays, and now we have Luminex assays, stem cell tools, proteome arrays, and many other tools. And then we have, of course, Tocris. If you're doing a Western blot, you probably are going to be working with cells upstream of that, and Tocris basically provides small molecules, inhibitors, agonists, antagonists that you would use as tools to interrogate and to say shut down the activity of an enzyme, a kinase, a phosphatase. You're looking at arresting the cells at a certain phase of the cell cycle, you'd use an inhibitor, or you're looking at an epigenetic change and looking at an HDAC inhibitor, or you're agonizing a GPCR receptor tyrosine kinase. These are the types of tools that Tocris provides. Then we have Protein Simple, and it's Protein Simple, by the way, very connected to Western blotting because they're offering and introducing really cool and innovative technology to replace Western blotting using capillary electrophoresis-based systems called Simple Western. And I'm going to show you a little information on that at the end. And also a new immunoassay technology where there's nothing like it called Simpleplex, which is a no-assay, highly reproducible, sensitive quantitation device for quantitating cytokines and chemokines. And then we have Novus Biologicals, which we brought into the family to really ramp up the number of antibodies that one can select from. And really, I'm going to have some information on this a little bit later, but really a really diverse antibody portfolio. Okay, enough on that. Let's move on here. So now, when I've given this talk before, I've gotten folks of different experiences with doing Westerns. I've gotten folks that have never done a Western, and then I've gotten folks that have been doing Westerns for 20 years. And really, this talk caters to everybody. So what we're going to do is, you'll have to forgive me for insulting the intelligence of anybody that's been doing Westerns, but we're going to do a quick review because I think it's important to cast a wide net. We're then going to get into transfer and detection optimization specifically, and then we'll have a little bit on choosing an antibody, and then I'll have a few other bits of information. I wanted to say a couple things just to start this off, which is, I've seen marketing data that has suggested that when someone purchases an antibody, the Western blot is the most popular application when using an antibody, even to this day. Now, the Western blot was pretty much introduced in the late 70s by a group led by Tobin et al. On the West Coast. I believe it was Stanford University. There's also another group that doesn't get as much press, but it's the #1 workhorse of protein research. And with all the sophisticated technology out there, most people are doing Westerns. Second thing I wanted to mention is if what you're doing now is working for you, and you're getting great signal and low noise, and your 150 kilodalton protein is transferring wonderfully, and your small 10 kilodalton low abundant protein is lighting up very nicely, and you're getting low background, you know what you do? You stick with what works. There's no reason why you have to follow everything that I'm suggesting. The purpose of this webinar really is about understanding the variables of Western blotting, and by understanding the variables, you're better able to troubleshoot and manipulate those variables to give you the best signal to noise. So you certainly don't have to do what I'm suggesting, it's just apply what you need. The third thing I would mention, and I'm kind of referencing this here, is we're going to do this talk in the order of transfer, optimization, followed by detection. Because think about it. If your protein is not out of the gel and onto that membrane and into the membrane, and by the way, proteins not only bind to the top of the membrane but into the membrane, you're never going to see it. Meaning you can use any antibody you like, you can change blocking agents and washing, but you got to make sure all that protein is out of the gel. And if you're dealing with a low abundant protein, say from the nucleus or primary cells, and you don't have enough protein on that membrane, you're never going to see it. So let's optimize detection first, and then a transfer first, and then we can follow that with detection. And that's how you really should troubleshoot your blot. Make sure all the protein transferred. So that's basically my opening segment here to kind of get the ball rolling here. So what I want to do is give you a quick review, and a lot of this is very familiar to folks. Now, if you'll notice, we have a section on preparing samples. Now, this could be a whole seminar, and I actually think there should be a seminar on sample prep because it's really, really critical. And at the end of the talk, I'm going to show you some pretty powerful data that suggests that it's not just the antibody that's the culprit in giving you a bad Western. It's garbage in, garbage out, as they say. So there's a lot of things you need to think about. We'll mention more of them at the end, but really you want to make sure that your lysates aren't old, that you minimize the freeze thaws, that your protease inhibitors and your phosphatase inhibitors are fresh, that you're catching the cells at the right passage number, the cell density is not too high. You might have to do an IP to enrich for that protein. A lot of people are using agarose beads for IP, and where you have to do a lot of pipetting of the beads, and sometimes you'll lose, when you're taking off your soups off the beads, you'll lose some beads, and that's protein you're losing. And so a lot of people move to magnetic beads. So that's another thing you could think about to try to maximize your protein recovery. Okay, so assuming you've done your sample prep, you then have to size fractionate those proteins, and the whole purpose of this is to separate based on size, though people have run Westerns on a 2D gel, believe it or not. It's a little bit messy, of course. You're going to size fractionate those proteins, and a question that someone may ask is, well, why can't we just add the antibody to the gel? Well, think about it. The protein is buried in that matrix of gel, and it's really about epitope access and steric hindrance, and that's the whole reason why people transferred the protein from the gel to the membrane to really just open up access to the epitopes and apply antibodies. And of course, once you've done the transfer, and we can do tank transfer and we do semi-dry transfer, and there's some fast transfer systems out there. And by the way, I think most people are using tank transfer and semi-dry systems. They're the workhorse pretty much, and there's PVDF and nitrocellulose, and I think the world is pretty much split. And as I had mentioned earlier, if you're using PVDF and getting great results, then you stick with it. If you're using nitrocellulose, then you stick with it. As we go through the talk, I'm going to point out some differences and some advantages and disadvantages. And then, of course, once we get the transfer, we want to do immunodetection. And there is basically three mechanisms. There's colorimetric, there's chemiluminescent, and fluorescent. And I mentioned that in the order of popularity, in the sense that, well, actually, to be honest, I mentioned the order of sensitivity. Most people, by the way, are using chemiluminescence because you can't strip and reprobe on colorimetric. Chemiluminescence is really the main workhorse, and you have HRP and AP. HRP tends to be a little less sensitive than AP, but most people use HRP because the signal is fast, it's sensitive, and it's sufficient. Now, fluorescence is not as popular, and part of the reason is you need some capital equipment, of course. Sometimes the membranes will autofluoresce, but multiplexing is possible fluorescence, and actually fluorescence is something a lot of people are thinking about because you can probe with multiple targets on the same blot using different colors, and that's the beauty of it. It's a little, and it's more sensitive, of course. Okay, the overall message is what? We need to have really great transfer. We got to get all that protein out of the membrane, out of the gel, and onto that membrane. And once it's out of the gel and onto that membrane, we then have to optimize our detection, and that's really what it's all about. So let's get right into protein transfer optimization. So I've got some variables here, and I want you to start thinking about these relationships, which is the size of the protein, right? The greater the mass of the protein, the larger the protein, the poorer the elution efficiency is from a gel. Meaning if I'm a large 150 kilodalton protein, my elution efficiency is very, very poor because I'm just so sterically hindered, I'm trapped in that gel. And that's why large proteins stay stuck in your gel. They're sterically hindered. It's not the only reason. I'm going to point another reason out a little bit later. But they're sterically hindered. So their elution efficiency is really poor. But their binding, once they get out of the gel and they bind to that membrane, their binding efficiency is really, really good. They don't have a problem with blow-through. I mean, the binding efficiency is really great with large proteins, poor with elution efficiency. For small proteins now, it's the opposite. So you got a 10 kilodalton protein. Its elution efficiency from the gel is really, really good. The problem is the binding efficiency. So because I'm a small 10 kilodalton protein, I don't have a lot of amino acids to create a bond with that nitrocellulose or PVDF substrate. So binding is really poor with small proteins. So we have two ends of the spectrum. Now related to that, of course, is your electron motive force, your current or the voltage. And so try to understand this relationship, which is the higher the current, so the larger the protein, the greater the current or the voltage you should use. Because what happens is, because it's basically like this, the greater the current or the voltage, the greater the elution efficiency is from a gel. So if I've got 150 kilodalton protein and I'm having trouble getting it out of the gel, I need to crank up my current and voltage to increase the efficiency and the force to get that protein out of there. But the opposite is true when it comes to small proteins, okay? Because if you're a small protein, you don't have trouble getting out of the gel. Your trouble comes when you're trying to bind to that membrane. So you typically want to turn the current or the voltage down. And that way you slow down the proteins, you slow down its speed of migration, and you give it more resonance time with the membrane to then create a bond. Okay? So it's really about, if you understand those variables, you can manipulate your current or voltage to really get things to either move out and get efficient binding. So there's a couple other things to think about. Obviously, so most proteins, by the way, are net negatively charged. And they have a lot of aspartic acids and glutamic acids, and about 70% of all proteins are net negatively charged. And they're going to run to red or run to the anode, and they're going to, you're using a typical Tobin's buffer, tris glycine, and they're not going to have any problem with moving in the right direction towards the membrane. There are some, rare examples, and by the way, these aren't common, but what they do is they illustrate the idea that charge has something to do with protein migration. So I was working in tech support several years ago, and I was talking to a researcher, and they had a very interesting situation where they were trying to transfer a 42 kilodalton protein, a 37, and a 33. They did the transfer, they did a detection, they were able to detect the 33 and the 42. It was the guy in the middle, the 37, that they didn't see on the membrane. Well, the first thing I did was I asked them how your transfer was, and I said, did you stay in the gel even afterwards? Well, it turned out that the protein, that 37, was still stuck in the gel. So the question is, well, why? Well, if you take the typical Tobin's buffer, tris glycine, which most people use, the pH of that buffer is about 8.2, 8.3, and most proteins, of course, are net negatively charged, and they're going to run to red, the anode. Well, this protein happened to have an isoelectric point at the pH of the transfer buffer. So what does that mean? Well, the isoelectric point is the point at which you have no net charge. It's the pH which you have no net charge. And if you don't have any charge, you're not going to move. So what was the answer? Well, the answer is you don't have to use Tobin's buffer. There are other transfer buffers. So I recommended what's called a CAPS buffer. It has a pH of 11. You shift the pH up, you give the protein a net negative charge, the protein moved. Again, not common, but just so you know, you can search the literature and find different transfer buffers. The other situation, and it's related to kind of cationic proteins, was that I had someone that was working with histones, and as you may know, if you're studying epigenetics, of course, histones are basic, positively charged proteins, highly positively charged. People are looking at post-translational modifications on the N-terminal tails, methylations, acetylations, and the like. Well, this protein is highly positively charged, so guess what direction it could potentially go in? It's not going to run to red, it's not going to run to the anode, it could potentially go in the opposite direction. And yes, I know there's SDS, which is your anionic detergent, but some of that gets washed away. So I simply had the person put the membrane on the opposite side to catch that protein. So the other thing I mentioned there, of course, is SDS, and SDS, of course, is very necessary. SDS can, of course, because it's a detergent, can affect protein binding to the membranes. Now, less so with nitrocellulose because of the mechanism of how nitrocellulose binds, and I'm going to describe that in a moment, but more so with PVDF, because PVDF is a hydrophobic interaction, and you're bringing in something that's very polar, and it can actually affect negatively the protein binding to the membrane. And I'm going to talk about this a little bit more coming up very soon. So let me just put an equation here, and I usually poll my audience when I give this live, and I ask folks, well, whose law is this? And I get like one or two people that will answer, and they'll say Ohm's law, and of course it's Ohm's law. So why do I have Ohm's law there? Well, I want to describe for you, this is basically the governing principle behind transfer, and it's very important. So look, you have tank transfer and you got semi-dry, and of course tank transfer, and years ago there used to be these huge tanks, right? Gallons of buffer, gallons of methanol, and for those of you that are old schoolers, they used to pour your own gels, use acrylamide, the Temid, and you degas the acrylamide. Now all the gels are reduced and they're precast, and of course it's more money now, but it's more convenient, of course. And tank transfer, of course, you can run in a half hour, you can run in 60 minutes, you can even do several hours and overnight. And then semi-dry came out a little bit later, of course, because people wanted to do transfer within an hour. So imagine this, you're doing tank transfer and you've got your buffer system, and what's happening over time, you're doing it for several hours, is that ions, you have a homogeneous ionic buffer environment. And so ions are starting to shift over time as current is flowing, as voltage is flowing. And what's happening is you're creating an asymmetric or non-homogeneous buffer environment. And so what happens, if you look at the equation, that's voltage equals current times resistance, is as the ions are shifting, you're not getting an even distribution, and thus what happens is resistance starts to go up. And as resistance goes up, what else goes up? Well, heat goes up. So this is the reason that a lot of people, or some people, will put an ice pack or put your tank in the cold room to minimize the impact of heat, because when you start to heat tris, your pH actually goes down, and if your pH goes down, guess what's going to happen? That's not good for proteins, right? We don't want protons floating all over the place and changing potential epitopes. So two things: keep it cold, and then you can sometimes put a little magnetic stirrer in the bottom of the tank and put it on a magnetic stir plate to keep the ionic environment mixed, homogeneous, and keep that resistance down and keep the heat down. Now, if you follow the equation here, what else is going to change? Well, as resistance goes up, voltage goes up, and as resistance goes up, current goes down. So this is why we don't want that, because I just mentioned about transfer efficiency and elution efficiency and binding efficiency to membranes. We want to run things at constant current, constant voltage to fight the effects of increasing resistance, and that's the bottom line here. Okay, so keep it cold, keep it mixed, and keep it constant. All right. Everybody uses methanol, right? 20%, maybe 10%, but I can almost guarantee you that most people don't know what methanol does. It's the necessary evil here. So let me just describe two things as to why you use methanol. And I'm going to give you the general rule here, which is, so look, if you took a gel, and this was more profound, of course, when you had poured your own gels and they're much bigger, but if you just use buffer without any methanol, and you had a gradient gel, you know what's going to happen? You're going to have this kind of asymmetric trapezoid type. you take the gel and you put it in the transfer buffer with no methanol, it's going to be very asymmetric, and it's not going to be very stable. So the first thing that methanol does is it stabilizes your gel, okay, because you want a nice stable environment for those proteins to transfer. So here's the general rules, and I actually wrote them down at the bottom of the slide here, which is the smaller the protein, the more methanol you use. The smaller the protein, the more methanol you use, say up to 20%. And why is that? Because the more methanol you use, the tighter the gel becomes. And we talked about elution efficiency. So small proteins, they don't have a problem coming out of the gel. Their problem is binding to the membrane. They can often do what's called blow-through. And thus, you never see your protein to begin with, even if your antibody is super great avidity, great affinity. So you tighten up the gel by using more methanol. It slows the protein down, gives it more resonance time, and more binding, better binding to the membrane. But what do you do with large proteins? You lower the methanol to, say, 10%. Why? Because the problem with large proteins, as we had mentioned, is they get trapped in the gel because they're sterically hindered. They're just so large. I'm 450 kilodalton protein. By the way, I'm going to show you a paper where they try to transfer a 480 kilodalton and how they manipulated things to get that to happen. But so by lowering the methanol, you expand the gel with more water, less methanol, and allow the pore structure to open up and allow the protein to come out. So that's the basic rule. And we're going to talk about membranes a little bit later and pore size, but basically to better capture a smaller protein, you want to use 0.22 membranes. And the reason for that is because it's thicker and there's a greater internal surface area, like 3 times as much as 0.45. So a general rule I tell people is anything 20 kilodaltons or greater, roughly. Again, everything's trial and error, but 20 kilodaltons or greater, you want to use 0.45. There's enough size there on the protein to get good binding, but anything lower than that, if you're working with a 10 kilodalton protein, guess what? You got to go 0.22 typically to better capture that protein there. And by the way, with 0.22 membranes, you typically get higher background. We're going to talk about that a little bit later. Now, the second thing that methanol typically tends to do is it tends to wash out the SDS. So as I mentioned earlier, nitrocellulose is not as affected by SDS, but when PVDF came out, it actually showed an interference with SDS. So, and by the way, PVDF is a great membrane, and I'm going to talk about PVDF a little bit later. But what you want to do is you want to take the gel. So after SDS page, you got your bromophenol blue tracking dye. You got a lot of unincorporated SDS and salts. You want to really soak the gel in the transfer buffer with the methanol to wash some of that SDS out. And that's going to get you better binding to the membrane at the end of the day. So maybe 5 minutes in the transfer buffer, then make your stack. And of course, when you make your transfer stack, you want to have it, you don't want to crush the gel. You want to get rid of all the air bubbles, because if there's any air there where your protein is, proteins don't travel through the air. I don't know if you know that or not, but they don't. So they've got to be in a buffer environment. So making a nice tight stack is really important. So that's the second thing that methanol does. And it basically cleanses your gel and gets it ready for prime time for your transfer. Okay, I kind of alluded to this, and maybe I jumped the gun here, but you've got 2 basic membranes. And by the way, if you didn't know, nitrocellulose, so nitrocellulose obviously has its advantages. It's the thing that started it all, and if you're getting great blots with nitrocellulose, you know what? You stick with it. It's basically a combinatorial binding mechanism, which is non-phobic hydrophobic interaction, non-polar interaction with those non-polar amino acids, tryptophanes, phenylalanines, isoleucines, and then there's a lot of dipole-dipole electrostatic interaction. And thus it uses both of those to get binding to happen. But with PVDF, of course, and by the way, nitrocellulose is brittle. It's not very flexible or pliable. It's also flammable, if you didn't know. It's not very chemically resistant, but it plays a major role in Western blotting. And again, stick with what works. And of course, you can do Southern blotting and Northern blotting, which I don't know if people are still doing, but it has that advantage because of its binding mechanism. Now with PVDF, of course, pretty much focused on protein blotting. It's very pliable. It's got great elasticity, right? So the handling properties are actually really good. Now its binding mechanism, of course, I usually poll my audience, how many people here is PVDF? And then I ask people, well, what's the first solution to put PVDF into? And of course, people will say 100% methanol. And the reason is because you're dealing with a phobic nonpolar substrate. You got to get that thing to be philic, polar. So typically you go into 100% methanol, and then 10, 15 seconds, and then you go a high purity water, millicue water, or whatever high purity water system you're using, and then into your transfer buffer. And you want to do that nice phase change to get it to be, it's almost like reverse phase chromatography. You've got to be in the same phase to get really great binding. I mentioned about, so by the way, what you're looking at here is the pore structure, the internal pore structure of membranes. And by the way, I know membranes are kind of, they're not very interesting. Membranes just kind of sit on the shelf and people just use them when they're needed. But really, the pore structure, when you do blocking of a membrane, you've got to block all of that. And so, by blocking all of that, you're really, there's a lot of real estate there to block. And so that's where all the proteins are binding. That's where all the antibodies are going. So you should have a real appreciation for what a membrane does, because it's really critical in this whole process here. I want to mention, too, that PVDF, if you're ever doing any kind of protein chemistry, PVDF was actually used a lot of the time in Edmond degradation sequencing because it can tolerate all of those organics. They used to do Edmond degradation sequencing right on the membrane, and it's also compatible with Kamasi blue stains because of the high methanol. So I want to show you this came from a paper, and these folks basically were the folks that introduced PVDF as an alternative substrate from Bio-techne, this paper that came out. And of course, nitrocellulose was the only game in town for years. And so they want to do a comparative analysis. And I think this is interesting data because, what it shows you is that not all things are created equal, and that what you do can really affect protein binding, because a lot of times we want to immediately blame the antibody for our Western woes, in the sense that we don't see our protein, so we say, oh, your antibody's no good. Well, not necessarily. The protein may have never been there. So, by the way, they show nylon here. Nylon's a very sticky membrane. I would never use it for Western blotting because I think you just get a ton of background. But look at what they did here. They basically radiolabeled protein and statically soaked these membranes in the radiolabeled protein. And then what they did was, just like in a Western, they incubated it in different blocking agents, 5% milk, 5% BSA, and then kind of as a control, PBS. But, if you look just under binding capacity, you can see that nitrocellulose actually binds a lot more protein. And so you may immediately just think, wow, I should just use nitrocellulose. But then after the blocking, look what happens to the percentages. They drop on nitrocellulose. They kind of are maintained, or at least you get better retention on PVDF. So, and if you look at just PBS, you're getting great retention on both. So what's the point of the slide? The point of the slide is that you're getting a little better retention on PVDF, and I usually mention this because if you're doing stripping and reprobing, you might want to think about this, because when you strip and reprobe that antibody, you're also taking off protein. But just simple things like washing and blocking solution can remove bound protein. So one little trick that I'm going to give you here, and I think this will work with nitrocellulose, but nitrocellulose of course is very brittle, so you got to be careful. Is after you do your transfer, take that membrane, okay, and I like to of course rinse the blot, in a nice high purity water, because you want to get rid of all the non-specific stuff that can cross-react and cause high background, of course. And if you didn't know, as a side note, pyrogens, sodium azide, EDTA, they all inhibit horseradish peroxidase, HRP. And if there's any of that potentially there, guess what's going to happen? You're not going to get a signal. So get rid of all that. And then once you've rinsed the membrane off after transfer, dry the membrane bone dry. And now when I say that, a lot of people will look at me when I'm in person, their eyes will get nice and big and they'll look at me like, really? And why am I saying that? Because what you're trying to do is, it's not a necessary step. It's just a potentially preventative step because you want to get as much protein bound to that membrane as possible. So like PVDF, you can simply put it at room temperature, put it on the bench, let it dry for a couple of hours, and then it's going to dry. And what's going to happen is, all those non-polar hydrophobic bonds are going to really firm up that bonding interaction because you're getting rid of all the water. And the same with nitrocellulose. You really want to just get maximum binding of that protein. Now, once that is done, of course, nitrocellulose, you can go right into your, TBST or blocking solution. But PVDF, of course, it's phobic now. You now then got to go back into 100% methanol, water, and then TBST to go then block. Because remember, you got to get into a polar state to get those blocking agents to penetrate the membrane, and those aqueous antibody solutions to penetrate that membrane and get to your protein. So again, the drying trick is just a way to potentially minimize protein loss, and that's it. Okay, so let me do a quick summary here. And the summary is this, and I want to focus on small proteins here. So I'm dealing with a 15 kilodalton protein. What are some of the things I can do? Well, I can definitely use 0.22 micron membranes. That's a necessity. I think SDS, especially with PVDF, has to be minimized because it's going to interfere with binding. And I'm a small little protein. I don't have a lot going for me when it comes to amino acids. So by minimizing the SDS, I want to basically get the best binding possible and not have that interference. And then, of course, I want to go low on the current and the voltage. Low and slow will prevent protein blow, basically. And, I might take the gel and rinse it in the transfer buffer, let methanol do its job. And then methanol, 20%. And that's a really great strategy for getting the best binding possible of a small protein. I wanted to mention something else. Oh, here. I'd forgotten that this paper was coming up next, but I want to conclude protein transfer with this paper that came, from the 80s, believe it or not. But the reason I like the paper is because it really kind of illustrates the multiple variables. As you can see here, and I'm sure a lot of you or some of you have had to do a large range of transfer, and it's quite impressive to be able to do this. And actually, I think they went up to 480 kilodaltons, and they use, by the way, tank transfer, they use nitrocellulose, and as you can see here, they use 20% methanol. And so this is what I'm thinking, is that, they wanted to basically use 20% methanol, and I don't think that they purposely used it thinking, oh, I want to, control my small protein transfer, so I'm going to increase the methanol. It might have just been, the recipe or the tradition of the lab. But they have also, so 20% methanol, as I mentioned, because, you want to squeeze the gel, tighten it up, slow down the speed of a small protein. But they add a little bit of SDS, and I'm kind of contradicting myself here because I said, well, you want to minimize SDS. Well, I referenced the fact that large proteins tend to get stuck in the gel and they don't transfer out because of their size, steric hindrance. Well, I think a second reason why they get trapped is, if you're in the presence of, say, methanol, say 20%, you can precipitate, crash out of solution if you're a large protein. And guess what the problem is? The problem is if you're not solvated, if you're not in solution, you're never going to move an electric field. So I think by adding a little bit of SDS, the purpose of which is to get those large proteins to resolubilize and get them caught up in the current or the electric field. And that way, you're going to get good transfer with these guys. And then what they did, and this references the electric field, is for the first hour they did really low current, and I think it was like 1 milliamp per centimeter squared or something. Again, low and slow to get those small proteins to come out. Not only to come out, they're going to come out no problem, because they're not sterically hindered, but to get them to bind. And then the next 16 to 20 hours, they ramped it up to 3 to 7 milliamps per centimeter squared, and to really force those large proteins to come out. So they did a two-phase transfer, and I believe they also doubled the tris and the glycine in the recipe for the Tobin's buffer. But anyways, the point is this: manipulate these variables that we discussed to get you the best transfer and the best binding to that membrane. And once you do that, then you're ready to go and all set for detection. Okay, that was my transfer portion of the talk here. So assuming that transfer worked wonderfully, we now want to move into immunodetection. And by the way, with immunodetection, of course, we could stain the blots. And of course, my quick comment on staining, of course, is there's reversible stains and there's irreversible stains. And the one message I will just say is if you're going to do downstream antibody-based immunodetection, you want to use a reversible stain, because if it's irreversible, it will block your epitopes permanently and you'll never be able to detect it with an antibody. So always use a reversible stain in combination with antibody-based detection. So what are the variables in immunodetection? We all know this, right? So you gotta find an appropriate blocking solution. Then you got your primary antibody, and hopefully they all come from R&D systems and Novus, of course. Ha ha ha. And I'm just kidding, of course. No, I'm not. But I will introduce antibodies from us in a moment or two. And then we've got, of course, consider the detection system. We've got, colorimetric, chemiluminescent, fluorescent, as we mentioned. So there are all these variables of wash solution. And of course, we use TBST, and of course, TBST has that tween 0.05%. And of course, tween is very necessary because what it does is it helps to clean all that non-specific interaction that you get with blotting, you know. And also, you know, when you're using antibodies, you know, antibodies can be sticky and they can tend to clump. And you want, you know, the best type of antibodies are when they're kind of, you know, single molecules and they can have better access to epitopes. And so it helps to keep antibodies from kind of sticking together. The FC regions can be a little sticky there. You know, there's a lot of hydrophobic domains there. So TBST, the tween is necessary, but I've actually seen some papers, believe it or not, that have removed the tween because it was starting to scrub off some of the proteins, believe it or not. But generally, people use TBST. Okay, let's go launch right into blocking. Now, again, a lot of this is trial and error. There's no, I'll just be honest with you, there's no magic bullet here, okay? And what works for one person and one antibody may not work for somebody else. So why do we block? Obviously, I showed you the pore structure of membrane. There's a lot to block there. We want to block the non-specific stuff, okay, because it's all about preventing noise. So we block, okay, and a lot of people will use, you know, all kinds of blocking agents. There's 3 to 5% non-fat dried milk, very, very common. And my comment on that is that you can use commercial sources from companies, or you can use something from the supermarket, Carnation or what have you. It really depends on your antibody system and what works best in your situation. It might be a little bit of a busy slide here, but I was trying to just collect my thoughts in terms of all of the different variables that one could manipulate. So first thing I'm going to tell you, if you've been using an antibody to P53 for years, the same antibody, and you've been using 3 to 5% non-fat dry milk, and you've been getting wonderfully low backgrounds, and then all of a sudden I'm now using an antibody against caspase-3, and I'm using non-fat dry milk, exact same 5%, and now I'm getting high background. Well, you know what you do? You change the blocking agent. You don't have to be married to non-fat dry milk. There's BSA, there's PEG, there's even non-protein-based, chemical-based blocking agents, which I actually think is a smart idea, because think about this. You've got your small 10 kilodalton low abundant protein that came from the nucleus or your primary cells, and you're trying to detect with your monoclonal looking for one epitope, and you've just dumped now a whole bunch of other proteins, caseins and the like from your non-fat dry milk on your blot. There's a great potential there that you're going to block your protein and epitope of interest. So it kind of makes sense to maybe try some non-protein-based blocking agents, because here's the thing, it's a balance. You're trying to not only block the non-specific stuff, but you also don't want to block the protein of interest. So some folks, and this would be my recommendation, that if you're having sensitivity issues, and I'm kind of addressing sensitivity here a little early, but you might want to lower the percentage of blocking agent so that you're not blocking all of your protein epitopes and you're exposing them to give you greater epitope access to your antibodies. And that might help with your sensitivity a little bit by not using always 5% non-fat dry milk. Again, every antibody in every situation and epitope is different, but the concept, I believe, is true. So that's one thing, which is change your blocking agent. Go to BSA. Second thing is, if you're making non-fat dry milk, make sure you, with a magnetic stir, you fully stir and dissolve that blocking agent. A lot of people might just take a 50 ml conical and shake it. I would absolutely dissolve it, because if it's not fully in solution, it's not going to fully block and be, you know, solvated to fully penetrate that membrane and get you good blocking. Of course, you may want to block longer. 0.22 membranes, by the way, are notorious for giving you higher background because there's a lot more internal surface area there. Do your washes, extra washes. Now here's a tip that I think, you know, I don't know if people do or they don't do, but that secondary antibody, typically a polyclonal, can be very powerful. And a lot of people use way too much. And I've seen great blots when, you know, at 1 to 20,000, 1 to 50,000, 1 to 100,000, because some of these ECL chemiluminescent substrates can be very powerful. So really, you know, a very simple step, and I think a very powerful step, is to dilute the secondary. You don't need that much secondary. Of course, lowering your exposure time, using a less sensitive ECL. I mentioned fluorescent detection. So anybody using, say, you know, an Odyssey system, these IR-based dyes, one thing to improve your signal is to read the membrane on the image or dry, and I think they tell you that. And then I believe they have a, their own blocking solution. But if you're using your own blocking solution, a lot of people will remove the tween from the last step because tween autofluoresces. And there are also, of course, low autofluorescing membranes out there you might want to investigate as well. Okay, and the last point, of course, is, if you're using a streptavidin biotin system, milk has biotin in it. So imagine what's going to happen to your blot. Okay. I'd like to just quickly address here, and we're slightly behind on our time here, so I want to just keep moving along here. So loading controls, and by the way, I went on Novus Biological's website, which I think is a really great site, and I'm not saying it because I come from Bio-techne, but it's really content rich. Not only does it have a great search engine, but it's got a lot of great useful information, and I saw a whole section on loading controls. I thought it was wonderful. So why do we use loading controls? Well, we want to obviously normalize our signal, and so that's the main purpose of it. We also want to make sure we're getting equal loads in all the wells of the gel. The other thing is people typically will check for the transfer efficiency across the whole blot. We just talked about transfer. Well, we want to make sure that we transfer everything equally across, because if you get a diminished signal, you don't want to think that could be a false negative for upregulation of a particular gene, when it could be that you didn't transfer very well to begin with. But the question I pose here is GAP-DH and beta-actin all there is? And the obvious answer is no. And so what I'm trying to illustrate here is, there's a couple of reasons I'm going to just tell you in a second, which is why you would not want to use GAP-DH or beta-actin. And what I'm illustrating on this slide here is that there are many other targets that one can use as your housekeeping gene, as your normalizer. And you should seriously investigate this. So one of the reasons is, you've got a protein of interest. It could potentially, of course, based on its molecular weight, fall at the same molecular weight as the housekeeping genes. And this would not be a good thing, obviously. So guess what? There are many others you can pick from. And here is a list of them. And they have the molecular weights there. Maybe simply pick something different that doesn't interfere based on molecular weight. The second thing that folks need to consider is the experimental treatment. So if I'm using a cytokine or a chemokine or some kind of protein for, say, R&D systems, and I'm treating my cells to stimulate them, which is very common for us. This is, people deal with R&D systems all the time for recombinant proteins. We work with them all the time. That recombinant protein treatment, or you're using a Tocris inhibitor, or using RNAi, or you've got some kind of mutation. The point is this: the experimental treatment can affect the expression of the housekeeping or the normalization gene. So you don't want to use it. So this is a very busy slide. I don't expect you to read it all, but like GAP-DH, it's affected by hypoxia. So you're studying the tumor microenvironment. You're looking at hypoxia. You probably don't want to use GAP-DH. Histone 3, if you're studying epigenetics, you're looking at post-translational Lamaze, you're doing a chip assay or what have you, they fall around 15 to 17 kilodaltons. You probably don't want to use that as a housekeeping gene. Okay? Let's get into antibodies here. And of course, antibodies are the key player, of course, in any immunoblotting. There's the primary and the secondary. The primary, of course, is probably the most important, and there's many different flavors, many different isotypes. Of course, you want to find an antibody that's been validated on a Western blot, and typically if the company, and like R&D Systems and Novus, we show you lots of data, and we show you data on lysates and not just recombinant proteins or peptides, which I think is a lot more powerful. Of course, you've got monoclonals, you've got polyclonals, and it really depends on the point of your experiment, what gives you the best immunoreactivity. Some people want to see multiple bands. Monoclonals, of course, go for one epitope. Polys go for multiple epitopes, tend to be more robust. I want to just quickly, because we're talking about antibodies, and Bio-techne, of course, is a major player in antibodies, and the two brands come to constitute probably, I would say, the largest selection of antibodies of any company in the world. 100% performance guarantee. I think what impresses me most is the diversity of targets. It's really one-stop shop, and 48 species. I mean, my gosh, that's a lot of species. I mean, we even have llama antibodies, if you can imagine, and alligator, and gosh, we have zebrafish, and I mean, it's just the list goes on. And for those of you that want to try an antibody, there are sample sizes. I know R&D has $99, 25 micrograms samples. But not only, the antibodies, and just to show you, and just one more slide on this, if you'll indulge me here, Really great search engine. There's publication, there's reviews, and on the left is a filtering system that helps you really dial down and get you exactly what you need. Okay, so what's my message on antibody titration? And again, the key word is titration. And again, there's no one way to do things, but here's a general rule of the game, which I would say that if you're having low signal, you should increase the concentration of that primary antibody, right? On the opposite spectrum, if you're getting multiple banding, right, and you're getting way too much, you want to decrease the concentration of the primary antibody. I'm going to show you some data in a moment. With secondaries, we already addressed this, which is, gosh, the first thing I would think about doing if I'm getting a high background is I would dilute that secondary antibody, and I think that's a really key step here. So let me show you some examples of doing some of this. Again, it's not easy. You know, you got to spend some time at different dilutions, but I think it's a necessary evil, if I may say so. But you've got to do it to get that sweet spot. So, by the way, you know, at R&D Systems, of course, we make antibodies, and we make 95% of all of our own antibodies. We're a major antibody supplier here. And to make an antibody, by the way, is not trivial. It takes years. It takes lots of validation and QC and screening. But this data comes from in-house, and I believe this is even online. But this shows you a very simple example where we block in 5% milk. So you've got blot A and blot B. The only difference here really is that we dropped the primary antibody concentration, and as you can see, we've really tightened things up, going from blot A to blot B. This is a polyclonal, by the way, and the milk solution has 5% milk and 0.15 molar NaCl. So a simple dilution can really clean things up. Here's another example. So we take a polyclonal antibody, and going from blot A to blot C, and forgive me, this may be a little busy, this slide, but basically going from A to C, we decrease the primary antibody concentration. We go from 1 to 0.3 to 0.1 micrograms per mil. But in this case, the primary antibody is in a 2% milk solution. Now, if we repeat this experiment below, the only thing different here is everything is exactly the same, 5% milk block, same salt, same antibody concentrations, but look what we do. We increase the percentage of milk in the antibody solution. So we've got multiple variables here that are affecting our signal and lowering our noise. And so by simply increasing the percentage of milk in the antibody, primary antibody solution, we're able, even from the get-go, to clean things up more efficiently. So you got to consider the percentage of milk in the primary antibody solution, the antibody concentration itself, and on this next example here, we're using exactly the same antibody concentration. We're blocking in 5% milk, but the difference is we're increasing the amount of salt, believe it or not, from 0.15 molar to 0.5 molar. And as you can see, there's an obvious result here, which is a much cleaner blot. Okay, so consider the salt concentration, the percentage of your milk in your antibody solution, and the antibody concentration itself. Okay, I want to just pivot a little bit quickly here and just talk a little bit more about antibodies in a general sense here, which is, of course, you know, when you're selecting an antibody, you often want to find the application that it was used in that particular application. Of course, you know, it's more powerful if you see data. So you know, at R&D Systems, you know, and Bio-techne, when we make antibodies, of course, you know, we have to screen. We literally screen them against all of these techniques, and of course, to be able to release them commercially, they have to perform. There has to be a high signal, and it has to be low noise. It has to be reproducible, and those that don't pass muster, they don't get released. And what works for one application will not always work for another, obviously. So, a couple things to think about, and I just want to take a few seconds on this. Of course, the first credible place that people look, of course, are publications. This is the obvious thing, and it's a real credibility statement when your colleague uses an antibody. But the second thing that I wanted to mention, which is, and we mentioned the word trust very early on in this talk, and you really should evaluate a vendor based on what they do to make that antibody. I'll give you one example. When making a polyclonal antibody, are they using a peptide or are they using a full-length, properly folded protein? Which is what we do because we make proteins. So that is something, because you want, you know, the most natural epitope state for that antibody to recognize, and not using peptides. Things like, you know, peptide blocking to assess specificity. And then there's one issue that has come up, I think, recently in the scientific community, and I think there's been some articles about this, and this is the whole idea of reproducibility and consistency. Because my gosh, if you run a blot on day 1 with an antibody and you can't reproduce that the second day, and a lot of papers, there have been some papers that have come out that have been questioned based on their reproducibility. You've got to be able to show lot-to-lot reproducibility with an antibody. It's really, really key. And this is internal data that this is actual data from our labs at R&D Systems Bio-techne that shows that from lot to lot we get the performance exactly right every time, perhaps plus or minus 10% CV. And this is a really important thing because you want to be able to trust that antibody in the long run, that it's going to perform lot after lot and you don't get any kind of drifting. Now, I want to show you some more powerful data. We talked very early on about lysates and sample prep. And this is a very underlooked area. And I got a lot of this data, by the way. I'd like to acknowledge Jeff Cooper, who is from our labs at Bio-techne, R&D Systems, who's in, I don't know, Jeff and I just met each other recently through emails, and he basically works in our antibody development group. And I thought some of this data was quite powerful because what it basically says is it's not always the antibody. We're always quick to judge the antibody. But again, garbage in, garbage out. So this is a very simple experiment that shows that, so what we did here is, and what we do is we screen, before we even screen antibody clones, we have to screen lysates because we need a optimal top performer lysate to then be used to then screen, say, our antibody clones. And so these are exactly the same treatment. These Dowdy cells are treated with interferon alpha exactly the same way. There were 2 different technicians involved, but the passages, they were collected at different passages, passage 42, 31, and passage 5. And what this shows is, this is a phosphostat 3 antibody, that you're getting different pre- and post-induction signal. And so, the question is, well, why? Well, there's multiple variables, and we started to talk about this. And I guess my overall message is you want to be consistent. You want to have a good positive control. You want to be consistent in your pipetting. You want to make sure your phosphatase inhibitors and your protease inhibitors are fresh and you're using the right ones. You want to minimize your freeze thaws. You want to, make sure your cell density is not too high and you're collecting the cells at the right passage number, and your reagents are fresh. And even, if you think about it, the protein quantitation assay you're using. So, you could be using a BCA, a Lowry, a Kamasi Blue, or, nanodrop, UV-based, and they all work based off amino acid content. You want to make sure you're consistent there because you can get different, total protein. And the point, the message of the slide is that don't always blame the antibody. Pay attention to your lysates. Another example, same exact conditions across, this is a phospho-FAC antibody, and showing exactly the same treatments, exactly the same cells, and we're getting the pre-induction is all very clean, but the post-induction gives you varying signals. And the question is why? Well, there's variations here potentially, and this was actually used to screen, potentially to screen, so they're doing the pre-screen in the lysates to then be used to screen clones. If you were to use lysate from lane 1, you might underestimate the power and the reactivity of that clone versus lysate 2, which shows a much stronger signal, and you might miss some clones that could have been very, very well, that could have been very, had great affinity and great avidity, versus using lysate 1. So you want to make sure your lysates are prepped properly and you pay attention to all the small details here. Another example, quickly, this is HeLa cells, PMA treated. Again, same conditions, just more data to reemphasize the fact that, identical treatments do not always give identical results. Pay attention to your lysates. Okay, I want to end the seminar, or the webinar, if you will, with a few things that I think are directly related to what we're talking about here. As you know, as we just got done discussing, Western blotting is not a trivial technique. It's a necessary technique. There's no doubt about it. But my gosh, it could take days, perhaps, to do a Western blot. And in the end of the day, you might not get data. I mean, the reality is sometimes things don't work out. So what I think is a nice complement to Western blotting is something is a technique, what we call proteome profile arrays. And this is kind of what I would consider a systems biology approach to doing protein identification and semi-quantitation. And by the way, Western blotting and these arrays are semi-quantitative, because really what you're doing is pixel density. There's no absolute quantitation here, as you may know. So when I say systems biology, it's a comparative analysis, this comparative look, because we tend to focus on one or two proteins. But really in biology, in the tumor microenvironment, and the signaling cascade, we're talking about multiple proteins that are all talking to each other and interacting. And to get a nice screen, a nice lay of the land, it'd be nice to look at multiple factors from the exact same sample and then follow that up with a Western blot. So let me just quickly introduce this concept to you, which I think could be a nice complement to a Western blot, say upstream of your Western blot. So this is a very simple experiment. A breast cancer cell line, T47D, I believe, and we grew them up, and then we treated them with EGFR. This is a phosphokinase array, and then we treated them with EGFR to stimulate the EGFR receptor. And then we used, and by the way, so what, I'm not even, I'll have to apologize. I'm not even describing how these membranes work. But basically, these are membranes that are coated with the capture antibodies already. There's no SDS page. There's no transfer. It's a nitrocellulose membrane spotted in duplicate with antibodies to multiple targets, intracellular or extracellular. You take the membrane, you grow up your cells, you treat your cells, you lyse the cells, you take the total protein, the total lysate, and you bathe the membrane in the lysate, rocking 4 degrees overnight. You wash, you add a reporter antibody, it's HRP on the end, you add ECL, and you get chemiluminescent Western blotting data, but without a gel. Hopefully that makes sense. And so this experiment is basically looking at EGF stimulation. We then add specific Tocris inhibitors to inhibit the activity of, as you can see on the right with the histogram there, of phospho-ERK, phospho-AKT at serine 473, phosphostat-3, and C-Jun. And this is nothing more than flatbed pixel density data, just like you process your Western blot. And the nice thing about this is it's a nice way to pre-screen to say, well, it looks interesting that, phospho-AKT is upregulated under this condition, and phospho-ERK is downregulated, and then maybe I should then follow this up and actually do a Western blot and get more specific. Okay, and I also wanted to mention too that not only can intracellular targets be probed, but you can also do cytokines and chemokines as well, and you would take cell culture soups or plasma or serum to do this. Again, it's a very, if you can run a Western blot in your lab, you can run a proteome profile array because there's no special equipment that's required. But it's just a very powerful tool. And I mentioned about reproducibility. If you're running multiple Western blots, you're running multiple samples, and inevitably you're going to have some variability there and lack of consistency. When you're looking at one sample, one technique, one shot, you minimize that variability. So that's another wonderful advantage. And you can go on our website, and there's multiple panels here, intracellular and extracellular, to target. Okay, last slide. We have, of course, our Protein Simple brand, and I wanted to just quickly mention, all of you, I presume, are doing Western blotting. So we have a whole series of really wonderful imagers now that can handle the membranes, can handle gel imaging. We can do colorimetric, chemiluminescent, fluorescent, really great software, flexibility, and imaging. And then down below that, and I referenced this a little bit earlier, Protein Simple is really revolutionizing protein quantitation and detection. And if you've not heard, this is called the Simple Western technology, and this is one of our systems, a very popular system. The beauty about this is there's no gel, there's no membrane. It uses very small amounts of sample. You can run about 25 samples in 3 hours. It's a capillary electrophoretic-based system, getting down at very sensitive nanogram levels, and if you're getting very quantitative, and I mentioned reproducibility, really great CVs on this system here. So I believe it's revolutionizing protein quantitation and analysis, and it's something that I believe is becoming more and more necessary because we just got done talking about Western blotting optimization, and as you can see, there are a lot of challenges there. This removes many of those challenges. So I would highly encourage you to investigate this. And as a company, we're certifying many of our antibodies on this platform. Well, I want to thank you all for attending this webinar, and I appreciate your patience. I tend to talk a lot. You managed to survive my webinar, and so I would like to open it up to Amanda, and to see if there are any questions out there. Thank you very much.
Thanks, Paul. That was an excellent presentation. And so we do have a few questions from the audience. Now, if anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So we have a question from Desiree. If your loading control is transferred with the same efficiency in the entire gel, can you extrapolate this to the entire gel or only for proteins that have a similar size as the loading control?
That's a good question. I would say that, because, we mentioned about the mass of the protein and the elution efficiency from a gel, I think you can only assume that proteins in the territory of size of that loading control transferred efficiently. I think if you're looking for proteins in the territory, the real estate of that protein size, then it's probably a safe bet that you got good transfer. And of course, I always tell folks to look at the markers, because markers can give you a good indication of transfer too. Though some markers, by the way, have dyes that are very hydrophilic and they can blow through the membrane sometimes. They don't get good binding. But if you're looking for a protein that's very large, I think, I would want to maybe perhaps check the gel. Like, after you do your transfer with the gel, you can actually stain the gel post-transfer and see if anything was left, in addition to checking the marker. So if you're looking for something that's really large or most distant from that size, I might want to just make sure that I got good transfer of that size. Because, like I said, not all proteins are created equal when it comes to transfer efficiency or elution efficiency and then binding efficiency. Hopefully that answers it.
No, that makes sense. I mean, that makes perfect sense to me. We got several questions pouring in. Okay, so this one is from, I'm sorry if I butcher your name. It is Maniselvin, and they ask, if they transfer for a long time, is it possible that the protein can run out of the membrane?
That's another good question. I've had this question before. I believe that once the protein, so if you look at the paper that I had referenced earlier, the Otter et al. Paper, they're obviously transferring an 8 kilodalton all the way up to 480, and they did this two-phase transfer, right? So they did the first phase at low Kerner voltage, I believe, to get those small proteins to bind, and then they cranked it up for like the next 16 to 20 hours. To get the large proteins out. Well, obviously they captured the small, they were able to detect the small proteins and the large proteins. So I think that's evidence right there that that continuing to run, because I think once the protein binds to the membrane, it's pretty stuck on there. There's a good interaction. And I think that paper illustrates very clearly that, running at a high Kerner voltage for a long period of time is not going to affect protein binding once that binding event has happened. So that's my answer there.
Yeah, that seems like pretty good evidence that that they're fairly well stuck on there.
Yeah. Yep.
And our next question is from Rebecca, and so she says that her lab uses PBST rather than TBST, and do they work as well as each other? Like, do both buffers work the same?
I've had this question before, and I believe I'm trying to remember. For some reason, if I can remember correctly, I think with signaling, looking at phosphorylation and these types of targets, I think TBST is preferred, but I cannot remember why. I think there are some targets where both will work equally well, but I really think it's based on the kind of target you're looking at, but I don't know the difference in terms of which targets are preferable over others. The signaling experiment, or the post-translational modification type of targets, is the one that comes to mind when it involves PBS, but I don't have any other examples. So I'm sorry, I don't have more information on that, but I believe in a perfect world, most of the times TBST is used, and there's a few exceptions where PBST would be used, but I don't know the exact examples. But I think just basically follow the protocols that are recommended for that particular antibody and that target.
Okay. And then we have a comment and a question from Juan. So he says, "Thank you. That was a nice webinar. And what he would like to know is whether proteins are able to renature in the membrane after the transfer. He said that he's read something about it recently, but it wasn't very clear to him. And I was kind of curious about this. I hadn't heard of that.
Oh, renature.
Yeah.
Yeah, that's a good question. I don't have any data to suggest it. I haven't read any papers. I could potentially see it happening because I mentioned the example about, and again, it's just a theory about why large proteins get trapped in the gel, and there's obviously when you're using methanol, of course, it's almost like cold acetone precipitation, right? So you're changing the nature of the protein, its structure. You're precipitating the protein such that something is going to fall out of solution and become just basically precipitate, crash out of solution. So I think it's very likely. I think it's very likely, especially in that organic environment. But I have not personally read any papers to suggest that. I'm wondering if there was a concern there on the transfer side of things, or perhaps in terms of detection, because if something renatures, it's possible that you're going to hide an epitope and not get an antibody to basically react with that protein once it gets to the membrane, in addition to having perhaps transfer problems. But no, I have not read anything in that arena, but I can imagine it happening, and I think it's quite possible.
Okay. And then Hikaru asks, what's your opinion on using total protein as a loading control instead of a housekeeping gene?
Yeah, I think total protein would be fine. I just think it has to be something that's consistent, and do all your checks. Make sure the protein concentration is accurate and your pipetting. But yeah, I just think as long as it's not affected by the experimental condition, and it's not obscuring the protein of interest in terms of molecular weight, it's kind of acting as an independent player. I don't think there should be an issue there as long as it's expressed equally across your lanes to show that you're loading the same and that perhaps your transfer is the same. Sure.
Great. And then Yvonne comments, says, great presentation, and asks if you have any advice for phosphoproteins, and specifically, how can she keep phospho groups in the membrane?
Well, phosphorylation, of course, can be tricky business because, you're dealing, it's interesting, I've seen data where, there was crude BSA used as a blocking agent and then very high pure blocking agent BSA, meaning the manufacturer of the BSA really cleaned it up versus, say, something for cell culture that's more crude. And in the crude blocking agent, of course, you have potential enzymes in there, and guess what which enzymes they are? Phosphatases and peroxidases. And guess what a phosphatase is going to do? So it's going to cleave your phosphate. So I think one thing to think about, of course, and I'm perhaps not addressing the question specifically here, but this thought came to mind, so I'm kind of addressing it. Is to make sure that your blocking agent is pure and it's a good quality blocking agent, so you don't have any issues. Because if you add ECL chemiluminescent reagent to the membrane, guess what's going to react with it? The membrane with all that blocking agent. And of course, there are phosphatases in here which can cleave phosphate groups. And a lot of times when you're doing phosphorylation, you're looking for one phosphate, which can be sterically hindered. So I think a good denaturation to really open up that protein, the boiling and the SDS. And then fresh, phosphatase inhibitors. And I say fresh, don't skimp. I don't know what inactivates phosphatase inhibitors, but I would make sure it's fresh, and then make sure you're using the appropriate ones that will give you the proper coverage and robustness of inhibition there. There was something else I wanted to mention too about non-fat dry milk. I think years ago I was dealing with an issue where someone was using a commercial non-fat dry milk versus, say, like carnation. We always see the carnation box on the shelf in a lab, and I think folks were getting better results with phosphorylation and detection using just the crude stuff, believe it or not. So I not all things are created equal. So again, I go back to the first thing I said in the entire presentation. Do what works best for you. You don't have to do exactly what I'm saying or what someone else is doing. Every situation is unique. hopefully that answers it. I kind of took a long route to try to answer it, but that's my answer.
I think that's pretty thorough. We have, and Wing contacted us before the webinar, and they had 4 questions. So, and they're kind of related, except for the last one. So the first question is, can you store the membrane after transfer and use it later?
Yes, you can. Yes, you can. In fact, there are protocols online. You know, my email is at the bottom there, and I can probably find you some of those protocols, but you absolutely can. You usually put the, now remember, PVDF is not brittle, so it's easier in terms of handling. We got to be very careful with nitrocellulose. There's actually that, I don't know what you call it. It's like the Wattman, I can't remember. It's like cardboard. You want to basically store it between 2 of those guys. And I'll have to, if you email me, I can find the reference for you on this. But there are ways to do it, and I believe you're going to put it at 4 degrees. And I've actually seen membranes stored even at -20, but 4 degrees is standard practice there. But if you email me, I can try and find some of those storage conditions for you.
Okay, and then this is kind of a 2-part question. So the first part is, how long does a purchased antibody last at 4 degrees? And can you keep antibodies in the freezer for long-term storage?
I don't know the answer to that exactly because I haven't seen the data. In fact, my colleagues at R&D in the lab would know this answer immediately, and they probably have a lot of experience with this. I think the freeze thaws are probably a factor because it's a protein. It's a glycoprotein, right? I don't know, to be honest. And I'm being very direct about this because I haven't seen data to know what the shelf life is in either condition. But to me, antibodies are proteins, and I would treat them like proteins. I just think that, they can break down like a protein can. They can denature like a protein can. So some of the same rules of the game can probably apply to them as well. And I mentioned, I showed you some data there, and we mentioned the whole topic of lot-to-lot consistency here. This is something you have to check because, things happen to proteins, to antibodies, and we want to make sure that the performance is exactly the same, or at least within the ballpark, 10% plus or minus on the CV.
Right. Yeah, I know anecdotally from my time in the lab that some of our proteins that were stored or not proteins, now that you've got me thinking about them as such, some of our antibodies that we stored at -20 didn't work after putting them in the freezer, but I don't know if that was because they were older or if it was because they were stored in the freezer.
Well, the other issue too is, now I think about it, is aggregation. They can kind of aggregate together, and so, I think that probably the best antibody is when they're in single molecule suspension, right? Like pretty much anything in, like flow, you don't want single cell suspension, right? So, I think, antibodies that are aggregated and sticking together will be probably less efficacious than, say, something that is, prepped in a way that they're a nice suspension together. There's this thing, known as the hook effect, which is like when you use too much antibody and they start to react with each other, and you'll see like the top end of a standard curve start to bend southward.
Right. Yeah.
So more is not always, more is less, or less is more, or something like that. I think less is more. Yeah. So more is not always better. Again, trial and error.
Okay, and then Shazia asks, is having problems with multiple bands even when they decrease the antibody concentration and increase the blocking agent. Do you have any other suggestions for something they might be able to try?
The old classic multiple band question. Yeah, that's like so common. Well, if you think about it biologically, it could be different isoforms. I don't know the exact situation, or splice variants. If you're using a polyclonal, one of the things, of course, we check for at R&D Systems is getting very specific reactivity. Like I showed you some blots. I don't know if it's the blot I'm thinking of, but even polyclonal, even though they recognize multiple epitopes, you can get reactivity on a single band, and we show this a lot. When we show polyclonal data. So I don't want to immediately just blame the vendor of the antibody. Hopefully she's not using one of our antibodies, of course. Oh boy, I'm in trouble now. but I would just say that you should check to see what the validation data is for that antibody, because it could be the antibody. Of course, I would say that other than that, I think just messing around with the antibody, I would ask, did she try to titrate the antibody? What was the percentage of blocking agent in the antibody solution? I might even increase the amount of salt. I don't know what her salt, molarity is in the antibody solution, but those are things that I think I would try. But dilution of that antibody, I think, is the most powerful thing to do. In addition, maybe perhaps use a less sensitive ECL reagent as well. So it might be take a combinatorial approach to minimizing those other bands. And there's the whole the last thing I'll say, I'm being a little wordy here, but is protein breakdown. I don't know if it's just individual unique bands or it's a smear, but sometimes proteins can break down, and thus you would get a smearing effect. But it sounds like more it's individual bands. So that's my answer, my non-answer answer.
Okay, so just to let everybody in our audience know, we've got about 5 or 6 questions left, and so we'll be wrapping things up after I ask those. So now we have from Hagar, this is regarding the mouse cytokine assay, and they want to know if it can be used for tissue samples and not plasma or blood or tissue culture.
Good question. Well, the arrays are validated on plasma, serum, cell culture soups, and homogenates. I believe cell and tissue. The answer is yes on tissue. I mean, you can technically use it on tissue. I don't know if it's validated for tissue, but you'd have to. I guess the question I have is, and I don't know exactly, but is the detergents that you're using, because so when we provide a kit, especially the intracellular kits, it comes with a lysis buffer that has a lysis detergent formulation. I believe there's a detergent in there, and they haven't told me what it is, and that has to be a certain concentration and it probably is some kind of non-ionic detergent, so it's not too harsh, so it's not going to interfere with the antibody-antigen interaction. But I would have to check with the lab. Now that I'm thinking about it, I have to check with the lab. But theoretically, it should be possible. The question would be, what is your detergent? Because these kits, and they are kits, are fine-tuned, and everything is meant to work together to optimize the signal and noise. Because to get these to work are not trivial. It takes years to pick the right antibody pairs and to create the best signal and the lowest noise and the cleanest background. So. In fact, my colleague Dave Finkel, who coordinated this, actually worked on a lot of these arrays in-house in our labs, would probably know if we've tried this in-house. But I would say theoretically, the long short answer is theoretically yes. Another approach I will suggest to you to do this that you most definitely can use to assess this is Luminex. R&D Systems has, I don't know if you're studying mouse or human, but this is a bead-based assay. It's actually quantitative. And it uses multiple standard curves from the same sample using 96-well plate. And you can most definitely assess in a picogram per mil fashion multiple protein quantitations, cytokines, chemokines, PDL1 from the same sample. So I think your approach, if you want to get quantitative on cytokines, is to try Luminex assays. And feel free to, I've run actually these assays many times, feel free to email me directly and I can point you in the right direction and describe more how these work. But basically, arrays are semi-quantitative. Luminex would be quantitative, and mainly for extracellular targets, but you can probe intracellularly as well.
Okay. And then Yeneth asks if increasing incubation times with primary antibodies would improve detection.
Increasingly, well, I think yes, but up to a point, because you think about it, you have epitopes that are exposed, you're going to get saturation. I think, the antibodies are all going to find their homes, and you're going to basically get a point of saturation, and I think you're going to max out. I just think it's, you've got so many epitopes for so many antibodies, and you're basically going to just bind up all those epitopes. And now, I bet you there's data probably in-house that's going to completely contradict me here, but at some point, you're going to get a robust signal that I don't think is going to increase much more unless you start getting a lot of, start getting more background, and you say, well, that's part of my band, and when it really is not. But and then of course you have to consider the ECL sensitivity of the ECL reagent, because that's going to enhance things even more. So the combination probably will give you your maximum saturation point.
Okay.
And exposure time too, of course.
Okay, and then Eric asks, when included, can the sonication during the preparation of the lysate damage some of the proteins?
I don't think so, other than the fact that you've got to keep it cold, because if you don't keep it cold, yeah, you can break down protein. But no, I think cold sonication is really the key here, because at the end of the day, you really just want the proteins to be denatured, unfolded, primary sequence of amino acids. And I think the only thing that could really, of course, you got to knock out those proteases. I know in-house we use, in our labs, we use hot, Jeff Cooper was telling me this is our lab guy from R&D. They use hot SDS, like 1%. And that knocks out all the proteases and the phosphatases right there. But I think heat at some point could potentially break down protein. So I think I would just keep it cold.
Okay. And then Katie asks if you know of an age-insensitive housekeeping protein that you'd recommend.
What sensitive?
An age-insensitive.
Oh, age-insensitive. I would say, if you go to one of my earlier slides here, let me just go back a little, if I may do this. I don't know if this is against the rules here, but I'm going to go back.
I think we'll allow it this once.
Well, okay, I love your flexibility. That's wonderful. Now, I'm showing the loading control page here, and I read something here about age, and I didn't. Let me see here. I bet you most of these are not affected, but there's probably one that is affected. Beta-actin may not be suitable for studies involving subjects with a large age difference. I don't know if that's exactly what she was referring to. I don't know the answer to her question. I would just say that probably most are not affected, but that's something you're going to have to just investigate a little more. But I've not had any experience in that area other than what you see here.
Okay, and then we've got 2 more questions left. So it looks like Megha asks, or Megha asks, in the proteome array, how can you recognize the proteins as there is no ladder?
Oh, in the proteome array, yeah. So this is a kind of a relative type of approach, and really what you're doing is you're not size fractionating the proteins via a gel and then transferring. So this is really, how can I put it? You've got a membrane, and they've got the capture antibody. Think of an ELISA, if you will, classic sandwich ELISA. You've got the antibodies, the capture antibodies, instead of the bottom of a 96 well on a plate, they're bound to the membrane. And so as that membrane is incubating in the lysate, in the cell lysate or the tumor lysate or soups or serum plasma, it's basically just capturing whatever's floating around, until you get an antibody-antigen interaction overnight 4 degrees. And thus, the antibody, that's the capture antibody, is the identifier. That's basically all it's doing is you're just identifying that protein based on its position. And there's actually a map that you lay down on these arrays and like a key to tell you which spot equates to what target, phosphostat 3, phospho-ERK, IL-6, TNF-alpha. And then you add, of course, a secondary, you form your sandwich, and then you get your chemiluminescent signal, which then you'll use pixel density. And the spots are in duplicate. And you take the average pixel density, and then that represents, and then you can graph that and all that. And then on the corners are positive controls. But really here, we're not talking about size fractionating and looking at things based on molecular weight. What you would do is, what I always recommend to folks is, look, you've got your research project. Go to our website, look at the panels of arrays, because they're going to have a preset menu for each array type. And see if those targets are relevant or impactful to your research. Like someone studying, apoptosis. And so that's pretty much an easy one. I send people to our apoptosis array, or they're looking at human pluripotent stem cells. Well, we have a human pluripotent stem cell array that looks at OCT4 and NANOG and some of these classic pluripotent stem cell markers. The kinases and the MAPK and the NF-kappa beta arrays, these are more specific, of course, but really it's just about looking at the targets that could potentially be of interest or could be related to what you're studying, and then selecting, the one that has those targets that are going to give you meaningful data. I'm studying cell stress. Well, maybe I should be looking at some heat shock proteins or MMPs, and cancer metastasis or whatever the particular area of interest is. And hopefully that makes sense.
That actually makes perfect sense. And now our last question is from Wynn, and I think this can be relevant for everybody. What's the best or most important advice for new researchers who have never done a Western blot?
Well, that's a very good question, and boy, I think it's the nice last question, because as I mentioned in the very beginning, I've gotten people that have attended this Western webinar or seminar, I typically give this live, that have never ever done a Western, and then there are people that have been doing it for 80 years, and everyone seems to learn something, because there's always a nugget or two that's new. And I would say that the best thing is to find there's protocols obviously, but there's no substitution for real live experience. Amanda and I were talking offline before this webinar, and she came from a biochemistry lab, and she ran tons of Westerns. Find somebody that's been running lots and lots of Westerns, that knows the tricks, because following a protocol or recipe is one thing, but to be able to work side by side with someone that can show you kind of the tricks of the trade and be able to do slight manipulations here and there is probably more powerful than just a protocol. And of course, gosh, these days you've got YouTube, you've got Joe videos, and they get really detailed. And by the way, the arrays, that we've been talking about, there's a nice video from the R&D Systems website that shows from our lab. You can even see what Dave Finkel, who works at R&D Systems, who helped develop these arrays, looks like, but it shows you exactly A to B to C, exactly how to do these arrays, and it's quite powerful. So I would utilize those types of tools, and that would be probably the best way to run a Western. Hook up with someone that really knows what they're doing.
Yeah, I agree. I think that you should never be afraid to bother the senior grad students or the postdocs or other people in the lab, or even calling up tech support. People love to help you.
Yes, and I wanted to mention one last thing about our tech support. And I know a lot of companies claim they have great tech support. We have wonderful tech support. And the nice thing about our tech support is that they're connected to the scientists that actually created the antibody, that created the recombinant protein, that developed the ELISA or the Luminex assay. And they have tons of validation data. In fact, I just demonstrated some of the data that we were able to provide folks. If you need an SDS page, or you need any kind of endotoxin information, they have just a ton of data to share with you, and they're willing to share with you and work with you to troubleshoot or make a product recommendation to get you better signal, lower noise, or whatever the issue is. So I give a great shout out to our tech support, both on the R&D Novus side and Protein Simple and Tocris.
Okay, well, on that note, that brings us to the end of the seminar. So thank you again, Paul, for a very illuminating presentation and a great discussion. I think you were a very big help to a lot of people. And thanks also to our sponsor, Bio-techne. And finally, thanks to you, our audience, for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours. There, you can also see the other webinars we have lined up for you in Bitesize Bio's Webinar Festival. So until next time, good luck in your research, and goodbye from all of us at Bio-techne and Bitesize Bio.